Well, hi everybody. Thank you for watching this recording. We're going to go over today a little bit about some genetic testing information. My name is Grace Snyder. I am a member of the University of Pennsylvania's Master of Science in Genetic Counseling program. And Gabrielle, I have had the opportunity to work with on this project for the past couple months, which has been fantastic. So what we're going to go over today specifically are some types of genetic testing and also talk a little bit about variants of uncertain significance. So this presentation was informed a lot by a couple of questions that we got from the Parental Advisory Board. And those two specific questions were, what are the different types of genetic tests? And then secondarily, what is a variant of uncertain significance in the context of a genetic test result? And to answer that question, we're also going to go into a little bit of information about what variants are, generally speaking. So diving right in, I'm going to talk a little bit about what the different types of genetic tests are. And to start, I'm going to give a little bit of background about genetics and DNA generally. So this might be familiar to you, it might not be, but what we know is that DNA is found in every cell in your body. It's made up of letters. And because it's made up of letters, you can think of DNA like a very long book. I like to think of it as an instruction manual for how to make a person. It's what makes you you. And what a genetic test is doing is it's actually reading your DNA to look for any changes in those letters that make up your DNA that could potentially impact your health. I'm going to talk about two concepts related to DNA before we dive into the specific types of tests. And the first thing I want to touch on is this idea of DNA to protein. So if you think about your DNA like an instruction manual, over here, let me get a laser pointer. This is your DNA, and it is instructions for how to make something in your body called protein. So the DNA in your body, since it's made up of letters, you can sometimes have errors in the letters that make up your DNA. And those errors will ultimately translate from the written material, so to speak, to these molecules called proteins. And proteins make up every part of your body. They make up your hair, your eyes, your feet, all of your organs, everything. So errors on this side in the instructions can ultimately lead to errors in protein. The other concept that I'm going to talk about is this idea of introns versus exons. And here in this picture, DNA is represented not as that twisted double helix structure, but it's instead represented as this long rectangle. And you can see in this picture that the DNA is broken up into two parts, these parts called introns and these parts called exons. You can think of exons as highlighted portions in the DNA book. So those are the really important parts and the parts that we're often most concerned about when we're doing any type of genetic testing. Not all the time, and I'll get into that a little bit, but if you think about exons just being this really important part, they are oftentimes where we're looking to see if there are any changes that could potentially cause Okerchung neurodevelopmental syndrome or another genetic disorder. And we know that variants in any part of the DNA, in either these introns, exons are more commonly what we see causing Okerchung. So we can talk now a little bit about what a genetic test actually does. So a genetic test is reading your DNA to look for letters, changes in the letters that make up your DNA. And in that way, you can kind of think of genetic tests like spell check for your DNA. And different types of genetic tests are going to be looking at different parts of your DNA. I'm pretty sure that I'm being recorded on camera, and if it helps, I actually have a physical cookbook here. So I might occasionally use this just as another visual example to help illustrate what these different genetic tests are looking for. So different types of genetic tests provide different information. They are looking at different parts of your DNA book. And I'm going to go over 4 types of genetic tests today. These are not all of the genetic tests that are available, but they are the ones that you'll probably most commonly encounter if you're doing any genetic testing for your child. So the first type is whole genome sequencing. Second type is whole exome sequencing. The third is a panel test or a multi-gene test. And the fourth is a chromosomal microarray. So to start, I'm going to go over 2 types of tests, whole genome sequencing or WGS and whole exome sequencing or WES, and compare and contrast them to each other. So whole genome sequencing is the most thorough of all of the tests. It is looking at every single part of your DNA. It's looking at every single word. And if we think about this little idea of introns and exons that I talked about before, it's looking at both of those elements. It's looking at both the introns and the exons, or those highlighted really important parts. So you could think about on this book, there's the title, obviously, but there's also like all these words up here. You have these testimonials on the back that I never read, but you might, someone else might read. And you have all of these, the inside cover, which just looks like the front cover repeated. And so you might not look at that. But whole genome sequencing is looking at all of that, all those words that you might not even think about. It's taking a peek at. Now compare that to whole exome sequencing. It's still looking at a lot, but it's looking at sort of the most important paragraphs in every chapter of the book. So whole exome sequencing is looking at the exons, and that's where the name comes from. Whole exome sequencing looks at exons. And if you use, again, sort of like the book that I have here as an example, you might not be looking at all these words up here or the testimonials on the back, but you're still going through and you're looking at all of the recipes, which is the majority of the book. That is what a lot of people would agree is sort of the most important thing to be looking at. Now, the interesting thing about whole exome sequencing as well is that even if you have a negative result on whole exome sequencing for Oker-Chung neurodevelopmental syndrome, it doesn't necessarily rule out Oker-Chung, because since we're looking at these highlighted important parts, there might still be something going on in these blue intronic parts that could be related to the condition as well. The next type of test that I'll talk about is a panel test or a multi-gene test. And this test is even more specific or targeted than whole exome sequencing. So panel tests are kind of looking at the most important words in a few paragraphs from like the really important chapters. And you can think of each word like a gene. I know we didn't define a gene earlier, but a gene is a unit of information that makes a protein. And these tests are looking, so if we look at this big picture here, if this is all of your DNA, it has all of the introns and exons. And then from that, we're taking the subset of the most important highlighted parts. And then from that subset, we're taking just a few of these pieces of DNA or those genes. That is what a panel test is looking at. So it's very targeted. And because it's so targeted, oftentimes panel tests are looking at very specific groups of conditions that are related to each other. So for example, a seizure panel test might, for the case of Oker-Chung neurodevelopmental syndrome, a panel test might be an autism or intellectual disability panel test that's looking very specifically at genes that we know are related to those conditions. Oops. And the last type of test I'll talk about is a chromosomal microarray. And this type of test is not like a spell check test. It's a little bit different. It's not looking at specific words or paragraphs. It's more looking for extra or missing chapters, so a bigger picture kind of test. And If we think of this book example of what our DNA typically looks like, when we're doing a chromosomal microarray test, we can see some results here that are a little bit different from what we're typically expecting to see. So in this case, chapter 1 of the book is duplicated. And on a chromosomal microarray test, that's going to show up as the color green. I have this little image here for anyone who's like interested in the science behind it, but don't worry if this picture doesn't make all that much sense. It's just to help illustrate that when we're looking for things, this test it's very visual, it's very color coded. And these are the actual colors that scientists see as results on this test. So like I said, chapter 1 here, it's duplicated, so that's going to show up as green on the test. Chapter 2 in this example, it looks typical, like what we would expect. It's not duplicated, it's not missing. So that's going to show up as yellow on this chromosomal microarray test. And then lastly, up here, chapter 3, or you could think of it as chapter 4 also, those are both missing. So either of those are going to show up as red on this test because it's missing. So that is another type of test that is not like spell check, but more looking at like big picture. Is there anything that is extra or missing in our DNA? So now that we've talked a little bit about what the different types of genetic tests are, we're going to talk about DNA variants. When you get a genetic test back, it's going to show you the variants that were found in your or your child's DNA. So I think it's useful to talk about this a little bit before we talk about specifically what a variant of uncertain significance means on a genetic test report. So we were just talking about these letter changes or these word changes in our DNA book, and those are called variants. So variant just means a change in the letters that make up our DNA. Genetic tests identify those variants. And I think what's important to remember as well is that everybody has a few variants in our DNA. It is what makes us unique. It's what makes our hair brown or our eyes a different color or makes us taller or shorter. So not all variants are harmful. The ones that are not harmful, we call those benign. And the ones that we talk about that can cause disorders like Oker-Chung neurodevelopmental syndrome or any other genetic disorder are called pathogenic. Pathogenic just means harmful or disease causing. So I'm going back a little bit to this picture I had before. So if we think about variants as changes in the letters of those instructions in our DNA book, then a change in the instruction here is going to lead to an error over here in those proteins. And like I said before, pathogenic variants are ones that can cause disease. They cause changes in those proteins that are important for certain parts of our developing bodies. So for instance, if you have a variant that affects a protein that is in your eyes, then you might have some sort of disease or disorder with your eyes. If you have a variant that is impacting protein that makes up your skin, maybe you might have some kind of skin issue, et cetera, et cetera. And again, pathogenic is just another way to say causing some kind of disease. So I'm going to go over 3 types of variants today. And all of the variants that I'm going to talk about are single letter changes in DNA. There are also multiple letter changes in DNA that are sometimes called deletions or duplications. But I'm going to focus on these 3 since we have a lot of patients in the registry who have these types of variants. So I know there's a lot in the slide. We're going to go through it together. But the first type of variant I'm going to talk about is a missense variant. And there are actually 178 patients as of the time of this recording who have a missense variant in the patient registry. Not all of them are necessarily the same variant, but I'm going to talk about one variant that at least a few patients have that is shown up here. And this whole text here is the name of the variant that you might see on a genetic test report. And this actually has 2 parts to it. So the first part is this C.593A to G part. And what that is referring to is the change in the letter in the DNA that is occurring. The second part, P.K198R, is the same thing essentially as this part, but it's just referring to the change that's happening in the protein part. That's what the P stands for. So all of the information on this slide is illustrating the same concept, just in a little bit few different ways, because some people learn visually and some people learn better with some text. So I'm going to start with this example sentence to help illustrate what's going on when you have a missense variant. So if we think about the sentence, the cat chased the rat. If you change one letter in the sentence, so for example, if you change this R to an H, that is changing the sentence, and it's changing the meaning or the sense of the sentence. And I've used a visual example here. So if this is what the DNA typically looks like, if you make a little change in just one letter, you can have this really big change where, wow, all of a sudden we're talking about a hat instead of a rat. And those are pretty different objects. I've tried to link that here to what's happening in the actual variants that you might see on a genetic test report. So here, the A represents what we typically expect to see, and that's getting changed to a G. And then also in this part here, that's showing the consequence of what's happening in the protein. Over here, I have it represented again, more on the scientific side. If this is what we expect the protein to normally look like, if this is sort of what the rat is in this example, then down here, this is what a change that we might see. We can see that it's a different shape. This is kind of like the hat in this example. So we can see how even a very small single letter change can have a big impact on how we read the DNA in our bodies. And some changes like that are very big, and some are more minor. If you think about maybe, for example, the H in the sentence being changed to an R, you would probably still be able to read the sentence and understand what it says mostly, and that would not be as big of an impact as in this case where we're having the whole subject of the sentence changing. The next type of variant I'm going to talk about is a nonsense variant. Now, I have a different variant on here. This is another name for a variant. This is a nonsense variant that we know causes Oker-Chung, and there are 13 patients in the registry as of this recording that have a nonsense variant. Not all of them are necessarily this nonsense variant, but this is one of them. And again, we have the C dot name and we have the P dot name referring to the DNA and the protein respectively. And in this type of variant, what's happening is you can think of it like if the original sentence is the cat chased the rat. In the altered sentence, there is a period that is coming too early. So things are being shortened a little bit. I've represented this visually here with the rat example as well. And then over here in the DNA, if we think about there being just a period too early in the sentence, that's going to lead to a protein that is a little bit shorter than we'd expect it to be. And the last type of variant I'm going to talk about is called a frameshift variant. It's kind of like a combination of the other two, so we'll go through that. In this type of variant, you have the major thing that's happening is a letter, in this example, is being deleted, but the spacing of all the remaining letters is staying the same. So in this case, the variant is C311del, and del stands for deletion in this case. So if we're looking at the sentence example again, here we're having a letter that's being deleted, but the spacing of all the other letters is staying the same. So it's just making it a little bit harder to read what's going on after that letter is deleted. And another consequence of the frameshift variant, similar to a nonsense variant, is that the period is coming a little bit earlier on in the sentence. So you both have a structural change where the protein is a little bit of a different shape. And then also it might be a little bit shorter than you expect because that period is coming just a little bit too early. There are 8 patients in the registry as of this recording that have a frameshift variant, some of which are the C.311del, but it's not the only variant, the only frameshift variant that you might see causing Oker-Chung. And so now that we've talked a little bit about what a variant of uncertain significance, or what a variant is, we can talk about what a variant of uncertain significance is. So when scientists first see these letter changes in our DNA or these variants, ideally they're trying to classify that variant into one of two categories, generally speaking, pathogenic or harmful, or benign or not harmful. And a variant of uncertain significance is kind of exactly what it sounds like. It's this middle category where scientists are uncertain what significance a variant has on your health. So when scientists do their classifications, they're using many different pieces of information, like pieces of a puzzle, to make that decision of whether a variant is pathogenic or benign. But if you're missing pieces of that puzzle or missing information, then you're going to be a bit uncertain about what category to definitively put a variant into. And that is how you end up with variants that have uncertain significance. And some pieces of information that scientists use to classify variants I've shown on the screen here. Some questions we might ask are things like, how common is a variant? If we see it in a lot of people who don't have a genetic disorder, we might say, okay, we don't think this is harmful. We think this is a benign variant. Something else that is important for scientists to know is how common Oker-Chung neurodevelopmental syndrome is, or whatever disorder they're studying. Knowing how common something is will impact the variant classification. Knowing the type of variant is extremely important. So nonsense, missense, and frame shift are just three types of variants that we talked about in the last couple of slides. So that information is actually very impactful in deciding if a variant is harmful or not. And then also understanding how the same variant, if it's seen in multiple family members, impacts those family members. So if multiple people in a family have a variant and they all have the same condition, we might be more likely to say that that variant is related to the condition that they have. So that's my presentation, a little bit about genetic testing types, variants, and variants of uncertain significance. I know the slide said questions. We had time for questions on the original Parental Advisory Board meeting. But if you do have any questions, please feel free to direct them to Gabrielle. And if possible, she can always pass on those questions to me. And I hope this presentation was helpful for your understanding. Thank you.